Today we're going to have a very interesting discussion about the medically important viruses. And these viruses are very common. Chances are you've had one of them, and they're widespread. They're around the world. Some of them, most of them, in fact, do have a vaccine as part of the treatment. So these viruses are important also in relationship to illustrating different principles of the field of virology and also immunity. So we will discuss those principles as we go through these viruses. So in terms of these medically important viruses, the question is, in what systems do they live, and how do those systems and principles about the viruses interact such that they stay around and they become, in some cases, pretty challenging to treat? So the example viruses that we're going to discuss today are the herpes virus, the papillomavirus, the rotavirus, measles virus, and influenza virus. Now, these viruses also differ in terms of whether they're DNA or RNA viruses. They differ in relationship to the site of entry and also, of course, the different tissues that they infect. So here's an overview of different viruses that infect the human body. And you can see all different kinds. You can see most particularly rabies, for instance. Now, the rabies virus is a virus that infects the brain, but it actually enters the body through an animal bite, for instance, and then travels to the brain. So as we've talked about in other lectures, some of these viruses infect the tissue that they enter, and some enter in one place in the body and then travel somewhere else. So here are the ones that we're actually going to talk about today. So we've got the influenza virus, of course, that infects the respiratory system. The rotavirus infects the digestive tract. The human papillomavirus is a sexually transmitted virus that lives in the epithelial cells of the cervix, or infects the epithelial cells of the cervix in women and the epithelial cells of the anus in men. Now, measles virus, they infect the skin cells, but they actually enter through the respiratory tract. And then a serious form of measles actually infects the brain. We also have herpes simplex type 1, which infects the oral cavity. So let's go through each one individually. The first one we're going to talk about is the herpes virus, Herpes simplex virus. It's a double-stranded DNA virus, and it primarily infects the epithelial mucosal cells. And the type 1 herpes simplex virus infects the oral cavity, and type 2 infects the genital tract. And they also can cross over, so we can see type 1 in the genital tract and then type 2 in the oral tract. The latency, so this virus does go through a latency period where it hides out, so to speak, in the nerves, and then reemerges at various points. Usually what causes reemergence is stress or immune suppression or some other reason, but they generally, for the most part, are dormant, or other folks call it latency. They're actually latent or dormant. And they occur worldwide, and they cause vesicles, or these mucosal ulcers. Are also known as cold sores, and the severe cases actually can cause encephalitis and meningitis, which is infection of the brain. Transmission is by direct contact, also through saliva, and also mother to child, and also through sexual contact as well. There's no vaccine for this virus. So the papillomavirus, probably many of you have heard about the papillomavirus because recently we've introduced a vaccine for young men and women. and it's a double-stranded DNA virus. Its primary site of infection are the epithelial cells again, and the epithelial cells of the skin and also the mucous membrane. This virus is also latent, so after the primary infection, the virus remains in the neurons, and it's a it occurs worldwide, and also it causes, initially it causes warts. All cases, I just read this in a manuscript earlier today in the literature. All cases of cervical cancer are thought to be caused by the human papillomavirus, which is why it's so important for our young men and women, adolescent girls and boys to get vaccinated, because after vaccination, you are protected from, girls are protected from cervical cancer and boys are protected from anal cancer. So there is an extremely high risk of malignancy if you've been infected, but a lot of people are infected and they don't get cancer. But if you look at all the folks who have gotten cervical cancer and trace it back, generally, in fact, in all cases, you can find some evidence of previous infection by HPV. So transmission is by sexual contact, indirect contact, and auto-inoculation, and quite a high prevalence of HPV infection occurs in the American population. So the vaccine that's available now is a vaccine against 4 different commonly occurring types of HPV. Now, the next virus we're going to talk about is a virus that infects the gastrointestinal tract. And this virus is also a DNA virus. And the primary site of infection are the epithelial cells of the upper 2/3 of the small intestine. And after replication in that area, the cells can actually, or the virions can be released into the digestive tract and then travel and infect other cells. Now, the rotavirus is, especially in the United States, is generally not deadly, but it can cause severe forms of diarrhea. And especially among young children, it's important to make sure that the child is adequately hydrated. It can cause release of quite a lot of fluids. So this virus is, of course, worldwide. It's very common in developing countries. It causes gastroenteritis. Now, the primary mode of transmission is fecal-oral, but also it can spread by direct contact, by shared contact, and very rarely through food. Vaccines are available for rotavirus, and it's especially important if you're going to be living in a country, for instance, that has a high prevalence of rotavirus, that you think about getting a vaccine. So here you can see the virus, and you can see inside the DNA is circular. And it has these different spikes, of course, on the outside. And those spikes are actually where the virus attaches in the intestinal tract. So these morphological attributes of these viruses are important in terms of attachment and penetration of the tissue in which it infects. So the next virus we're going to discuss is the measles virus. Now, the measles virus is not a DNA virus. It's actually an RNA virus, so it's single-stranded. And in this case, it's one linear strand. Okay, it occurs worldwide, of course. Now, the incidence of measles in the United States has gone way down because, number one reason, we do have a vaccine for the measles virus. And it's important that, especially in today's world, that mothers think about vaccinating their kids, because if you don't vaccinate your kid, for instance, then you could be a source of infection for other kids. And often unvaccinated children live amongst children who are vaccinated, and then they don't get the disease, but it's because everybody else around them are vaccinated. So that's always been a big controversial discussion about whether mothers certainly want to vaccinate their children, but it's very, very important, not only for their own children, but for other children as well. So the measles virus, of course, causes a rash, but it's transmitted by an aerosol, by airborne droplets. And the measles virus initially enters the body through the respiratory tract. And of course, vaccines are available. And you can see here we have the same attachment, I guess, organelles, so to speak, or the same attachment structures that viruses have that help them implant in terms of the respiratory tract. And so the final virus we're going to talk about is the one that's often on many people's minds, especially in the wintertime, is the influenza virus. And this virus actually has very characteristic spikes on the outside of the virus. And it's an enveloped virus. And the envelope, of course, consists of a lipid bilayer. And there are two types of spikes that occur on the influenza virus, and the hemoglobin or hemagglutinin, spikes, and the neuroaminidase. Sorry, my pronunciation at times needs some work. And these two different kinds of spikes are what's responsible for the differences that we observe in viruses. Now, viruses, because they're RNA in nature, it's much easier for them to have mutations and also to recombine. So in terms of developing vaccines, we continually, at least on a yearly basis, need to revise the vaccines to reflect the new changed influenza viruses that are found. So there are actually 16 types of these HA spikes and 9 types of NA, and the particular combination of these is what determines the different strains of influenza. So the one actually shown here is called H5N1. So it's the 5th type of the HA type of spike and the first type of the NA spike. So the concept, antigenic drift, is very important in relationship to influenza, and that's the concept where these spikes just change ever so slightly from year to year. And actually in individual amino acid bases, now those spikes are composed of protein, do change, and when they change, they cause what's called antigenic changes. And that concept of antigen or antigenic refers to the immune system capacity to recognize it. So when you have these slight changes that occur in the antigenic shape of the influenza, then the immune system needs to relearn the shape. And if the shape is different enough, then the immune system won't recognize it when it's infected by that particular virus. So these changes can even occur within the influenza season. So sometimes people complain, oh, I got an influenza virus and then I got influenza. What's wrong with this vaccination? Well, actually what can happen is you can get a vaccination and then you can get infected by just a slightly different version. And even getting infected by that slightly different version, you'll still get sick. But often, in fact, most often, the second infection that you do get within that flu season is much milder than if you wouldn't have gotten vaccinated at all. So these new influenza strains are continually emerging, and as they emerge, the vaccine seems to be less effective. So, that's why, for instance, folks can get two influenza infections in one season because potentially the second infection is a different type of influenza. So there is some crossover between the antibodies of the two types. So fortunately, or usually, that second infection is not as serious as the first. But these antigenic drifts, cause these epidemics, or these seasonal epidemics. Now, when these strains actually reassort, then you can get a much more extreme rearrangement. And then when that's called an antigenic shift, which means that the change is much greater than what's usually seen on a year-to-year basis. And that's when the CDC, for instance, gets very concerned. So we had an example of this in 2009 with the H1N1 virus, and everyone was very concerned. We, had all our, at least here in California, we had all of our hospitals ready for this pandemic, and the pandemic never really occurred. But you can't go back and assume that it wouldn't have occurred anyway if you didn't vaccinate everyone. So certainly a lot of folks here were vaccinated, and the epidemic was comparatively mild. Right now, we're also being concerned about this H5N1, which is another version. And so many folks this year, for instance, got a combination, myself included, got a combination influenza vaccine to potentially protect against all these different types. So often the way in which we get these extreme recombinations is by actually recombining with influenza that are found in other species, most often in birds, but also in swine, or other folks call them pigs. We get these recombined viruses that recombine with human viruses, and then they become a brand new version that we have not seen before. And those are usually very pathogenic, which means it's very likely that folks will get sick from those viruses. So these new strains actually leaves an entire population vulnerable, because the population has never experienced that particular virus before. No one's immune system has a memory of any kind of infection like that. And so everyone in the population is potentially susceptible. So with H1N1 that occurred in 2009, once the CDC and other places and the WHO around the world got wind of this potential pandemic, there was a huge influx of bioengineering, and a lot of scientists got together, and they were able to reconfigure the vaccine in less than 6 months. I mean, it was pretty amazing. And then they were all these different all the vaccines were flown all over the world, and there was quite a lot of immunizations. And then H1N1 did not prove to be that big of a deal. So these antigenic shifts contribute to this disease pandemic, and it's really because of this dramatic recombination that often occurs with other species. So here's another picture of that. So here is a picture of how this all can occur, where we have an avian virus that infects birds, influenza virus, particular virus that infects birds, and then pigs are what they call a mixer species, and so they're a particular species that can harbor both avian viruses and human viruses. And within the pig, you can get these types of recombination that couldn't occur in humans or in birds. So we get this mixing of different viral types into a brand new virus that can be very, very pathogenic in relationship to human beings. So many of you have probably, or most of you have heard about the flu pandemic that occurred in the early 20th century, and up to 50 million people died from that virus. And it was because, and actually it was also an H1N1 viral type, and the population, it was the same thing. They had never experienced H1N1 before. We had no, our medical technology at the time was obviously not well developed, so quite a lot of people died from that. So when H1N1 came back in 2009, remarkably, or understandably actually, we needed to be prepared for that one. So to conclude our discussion, let's have our questions. So the first question, or the first example, has to do with what system does the measles virus originally infect? So we've had this discussion a couple of times throughout our viral lectures, and it's the concept that the virus can infect or can enter the body in one place and live somewhere else. And a lot of the principles of natural selection also apply within the human body. So in terms of natural selection over time, the virus, the measles virus, seemed to do best in the epithelial cells of the skin. And that's where you see the characteristic rash of the measles virus. But folks think, oh, well, that's how you catch measles, is from, touching someone else who has measles, or, skin to skin contact. And that's absolutely not true. The virus is spread through air droplets into the respiratory system. And then we know also that a very serious form of measles can occur in the brain, an encephalitis. And that is very serious, and that's certainly something that we don't want to have. And that's also the reason to vaccinate your kids, is to prevent that possibility from happening. So our second example has to do with the herpes simplex type 1 virus, and what does the herpes simplex virus share in common with the human papillomavirus? Well, type 1 and type 2, remember type 2 as well. We have two types, and then we have HPV. So all three, in fact, are DNA viruses. Okay, and they both infect mucosal tissue. Okay. At least herpes simplex type 2 is sexually transmitted, but type 1 can be sexually transmitted as well. And of course, so is HPV. And the other thing they share in common is that they're latent, so that they actually hide out in the neurons. So those are the ways those particular viruses share things in common. And then, of course, there are differences as well. Now, the herpes simplex viruses do not have a vaccine, whereas, of course, HPV does. So our last example has to do with the concepts of antigenic drift and shift pertain to the need for yearly vaccinations. Now, we did discuss that today. And it has to do with, because influenza is an RNA virus, and we learned in our microbial genetics lecture that RNA viruses are actually more prone to recombination and mutation. And so because of that, there's an increased likelihood that viruses will change over time. Now, another prime example of an RNA virus, by the way, is HIV. And one of the reasons why we don't yet have a vaccine for HIV is because it's constantly changing. It's very challenging to develop vaccines for viruses that are changing shape all the time, because you develop it for one shape and then it changes. Now, in the case of influenza, thankfully, we have change there as well, but it doesn't change as quickly, and it tends to change slightly on a yearly basis. So we're pretty good. It's a pretty good solution to develop these yearly vaccines that everyone tends to get. I mean, the best time to really get them is in the late fall, early winter, because usually influenza starts in the early to mid-winter months. So anyway, thank you so much for visiting educator.com.